So we have exam on Tuesday?
No.
I mean the following Tuesday.
No, the one after that.
All right. Yeah. And review is on Saturday, next Saturday, not this Saturday, but next Saturday. Okay. All right. The 5th, yeah. The 5th. So. All right. No questions? Oh, ribosome. 70S and 80S. What do you think, David? You tell me? Okay. All right. Okay. All right.
Can't wait to hear this one.
I didn't look it up, but from last time,
Yeah.
I thought about it, and it's probably some of what dissolved.
Some of what dissolves, yeah, okay. One of the answers is because when they unite, some of the ribosomes dissolve. Okay? Okay. Oh. But did I say that's the right answer?
No.
Okay. Okay, all right.
Oh, okay.
That's one of yeah. That's one of the answers I have received, which is unfortunately not the right answer. Okay. But yeah, okay. All right. Who was going to give me the right answer? Go ahead. What does that mean? All right. Okay. That's exactly what's the answer. Okay. One word answer is hydrodynamics. What happens is this is 30, a small portion of the ribosome, right? Okay. Okay. It has nothing to do with really centrifuge and all that you read in the book. The function of the ribosome is what? Protein synthesis. But this is what they observe in the lab. When scientists, they do research, they find out these things accidentally. And this really doesn't have much practical use, okay? Because the protein, the ribosomes, they produce or they synthesize proteins. Okay. So when they were probably studying protein synthesis, they observed that when these two parts are separate, small part and the large parts, when they sink in liquid, let's say the small part takes 30 seconds to sink to the bottom and the large part takes 50 seconds. Here's the small part, here's the large part. Separately, they have different shapes, different shape. The answer has to do with the shape of the ribosome. So when you put them together, they become more hydrodynamic. They sink faster. That's it.
What about the loss?
Oh.
I think it was a UKS is 60-40?
No, 50-40. 50-40. 50-40.
So where do you lose that 10?
Oh, the sink faster mean the weight is not being lost. It's just the 10 second is lost. The 10 seconds faster, they will sink 10 seconds faster. There's nothing being disintegrated or lost. The time of travel, the time of sedimentation is 10 seconds faster. That's what it is.
So
yeah.
that's it. Okay, so that's so the 70 and 80 is time of
sedimentation.
Time of
sedimentation, correct, yeah. Sievberg unit, yeah, they sink faster. That's because when they're together, their shape changes. Their shape changes and they sink faster. That's why you have -10. Separate, their shape is different. Yes, Jackie?
Yeah, the question number 4 in the pretest.
Yep.
I didn't really understand it. It said if you add 4 cups of distilled water to a 2 gallon.
Yeah, we have not talked about that yet. Yeah. We have, they are about 70% of the exam we have not talked about. It has to do with the cell wall. We are going to, as soon as we do that, you'll have, sure. Yeah. Any other questions? We finished chapter 3. We started chapter 4? Okay. All right. So let's continue with chapter 4. Hopefully we'll finish maybe half of chapter 4 today. All right. While it's warming up, let me ask you a few questions. Okay. Fast learner. Fast learner. Yeah. All right. Let's see. Not necessarily. This is one of the scenario type questions that you'll have on the answer. On the test, scenario type. You went to, let's say, South America for your summer vacation, and you collected some water samples from one of the rivers there. And you brought it to the microbiology lab, and you're doing chemical analysis on the bacteria that you found in the lab. This is one of the things that you found in that bacteria, that it cannot convert CO2 and water to glucose. Number two, that you found out that this bacteria has DNA only. Okay. Third thing, tons of PG, peptidoglycan. Lots of peptidoglycan. Okay. What else can I ask you? All right, let's see. Now, for now, whatever we have learned. Okay. First of all, you want to observe intracellular structure of this organism. What type of microscopy would you use? Intracellular structures. Okay. You want to observe the intracellular structure of this brand new microscope that you have found in the water sample that you have just brought from South America. What type of microscopy would you use to observe the intracellular structure of this microorganism? Bright field. I didn't say living. Did I say living? No. Yeah. You can say electron. Sure, acceptable, absolutely. If you have electron, sure. But start with simple bright field. Bright field. Bright field, how about phase contrast? Phase contrast, absolutely. Phase contrast and electron. Can you use fluorescence? No, can't do that. Dark field? No, can't go inside the cell. All right. Is this bacterium gram-positive or gram-negative? Which of the characteristics? Tons of PG. This tells you that this organism is gram-positive. Okay. Is this autotrophic or heterotrophic? Okay. Remember the term we talked about? Autotrophic and heterotrophic. We did not?
You were supposed to do it.
Okay.
Yeah, you just
No problem, sure. Okay, so I must do that before the exam. So let's do that today. Okay, okay, all right. Okay. This is a heterotrophic organism. A heterotrophic organism. Let me write this down. Heterotrophic and organisms that are autotrophic. Okay. The definition of heterotroph first. Cannot cannot convert inorganic compounds such as carbon dioxide and water to organic compounds such as glucose. One more time. Heterotrophic organisms are those organisms that cannot convert inorganic compounds to organic compounds. What are we? Are we autotrophic or are we heterotrophic? Oh, so you can go to the beach and absorb the sunlight and make your own food? Unfortunately, can't do that. If all of us could do that, no one will be sitting here, believe me. We are heterotroph. You need your KFC and pizza and your steak and all that. Pre-synthesized organic compound. We need organism. They cannot change. You cannot just breathe in carbon dioxide and drink water and lie down on the beach and make your food. On the other hand, have you ever seen a plant going to Winn-Dixie? No. Autotrophs. Autotrophs, they can convert. They can. And this process in which inorganic compounds are converted into organic is called? Very good. Photosynthesis. We do the opposite. We human beings and most bacteria, we start with organic compound and we break it down into carbon dioxide and water. And what is that process called? That is called cellular respiration, in which glucose is broken down into carbon dioxide, water, and ATP. Cellular respiration. Okay? So heterotrophs, they are responsible for cellular respiration. Which is simply ATP synthesis, energy synthesis. Okay? All right. So now let me come back to my question. Is this organism autotrophic or heterotrophic? Heterotrophic. Okay. Okay. Third and last question, then we'll move on. Okay. Is this organism prokaryotic or eukaryotic?
Prokaryotic.
How can you tell?
DNA.
DNA only. But you really cannot tell on the basis of this one, because both prokaryotes and eukaryotes, they have DNA.
In your question, when you say DNA only,
Yep.
isn't that telling you that it's...
But you really cannot tell. Yep. See, if it says DNA only, you can't tell autotroph. DNA only, okay? Or if it says DNA only, you cannot tell prokaryotes or eukaryotes. Okay. I need to elaborate. Yeah, this is not a good question. Yeah, I need to say more about that. Yeah. I will say DNA in a nuclear membrane or DNA without nuclear membrane. Now that will be more clear. Okay. So DNA only, you will have a choice. If I have this, it will say cannot answer or none of the above. Okay. Yeah. And there will be lots of questions like that on the test. Yeah. Don't worry about it. Okay. Let's move on. Okay. Okay. Chapter number 4. Everyone received all the information that I bombarded you with all the information yesterday, right? A whole lot of information. Yeah. Okay, here we go. We finished that one? Oh, what is this? Holy mackerel. Okay. Did we talk about this or no?
No.
Two, we did not. Okay. All right, 2. Let's start with the similarity, just to review. Orlando, please. Thanks. Last time when we left, we were talking about the differences. Thank you. Similarities between the prokaryotes and the eukaryotes. Okay. We talked about all these. Did I tell you the functions of all these, right?
Yep.
Okay. We both carry out, because reason we carry out chemical reaction is for what? To metabolize food for what? Because we are heterotrophs. We need organic food for the purpose of energy. Food to basically get ATP. Okay? So most bacteria are here, right here. There are a few bacteria that are here, and you can name them. They are cyanobacteria, blue-green algae. Cyanobacteria, they belong to autotrophic. Cyanobacteria.
Just so I'm clear, autotrophic are prokaryote? What did you say about
No. Two different categories.
Okay, but
Yeah.
where did you go with bacteria?
Okay.
Autotrophic.
Most bacteria are here. Most bacteria, they need food. You need petri dish to give them food. Okay? You have to give them food to grow. But some bacteria, they can make their own food, like cyanobacteria. They're all bacteria, but most bacteria cannot make their own food. Some bacteria, they can make their food. Synthesize proteins. We both, prokaryotes and eukaryotes. Let's name the three most important functions of proteins. Number one. Cell synthesis. New cell synthesis. Good. New cell and cell components. And number two? Enzymes. All metabolic activities. Okay? They cannot take place without enzymes. And all enzymes are proteins. But not all proteins are enzymes. All enzymes are proteins. You cannot have a metabolic activity without enzyme, anabolic or catabolic. But all enzymes are protein, but not all proteins are enzymes. Third function, there are more. Antibodies. Very good. Antibodies. All antibodies. 5 major groups. All of them, they are protein. Protein. Store energy. Both prokaryotes and eukaryotes, we store in the form of what? Basically ATP. ATP. Some bacteria do actually store glycogen. Like me, I store right here, you see? Right here, okay. Fat or glycogen, or if you are athletic person, you store it in the form of glycogen in your muscles. Okay. If you're a couch potato, fat. Okay, like me. Okay. Come on. Thank you. This is pure memorization now. Shapes, size, and arrangement. Most bacteria that you encounter every single day, they exist in 3 shapes. They are what? Round, rod, and spiral. Chain will be arrangement. Yeah. Round, rod, and spiral. Those are the 3 basic shapes. Okay. Basic. I told you, bacilli. This can cause some confusion sometimes. Bacilli. Okay. Bacilli could be shape and the name of the bacteria. Not sure if I mentioned that to you or not. I did. Okay. So be careful. So if you want to mention the shape, use the small b. If you want to write the name, always write with capital B. Capital. Okay. Single. Coccus, singular. Cocci, plural, round, spherical. Some arrangement we'll talk about later. Okay. Some, they always grow in pairs. Some, they grow in chain and so on. Shape. All right. Okay. Third, spiral. There are 3 subcategories of spiral-shaped bacteria. The first spiral shape is called Vibrio, which are causing big trouble in Haiti. Cholera. This is the bacteria, that banana-shaped, sausage-shaped bacteria, comma-shaped bacteria. This is the one that causes cholera. Okay? Called Vibrio cholerae. This one right here. It could be deadly if the person not rehydrated. Okay, it causes extreme diarrhea and vomiting. Okay, if the person is not rehydrated, the person can actually pass away. Second, spirula, plural. Singular is spirulum. Check for the spelling. I think the spelling may not be correct in your notes, so please. Spirulum. Oh, sorry. L-U-M. L. I thought I. L-U-M. Spirulum. Okay. Spiral shape. Two things that are important here that I have put in red words here, letters. Their bodies are rigid. They're motile by polar flagella. Polar flagella. What is polar flagella? Flagella that is attached at one end or both ends. It could be one end or both ends. Okay, that's called North Pole, South Pole, right? Okay, so either end, that's called polar flagella, here or here. All right. We will look at the different types of flagella later, but polar means end, terminal. Sometimes they are also referred to as terminal flagella, terminal. Terminal or polar, same thing, terminal. Spirulum minor. If you eat rats, people do, okay? Or if you live in New York and travel on subways, your chance of being bitten by rats are greater, okay? Careful, this lives in the mouth of rats, okay? I don't know if you watch the Dirty Dining on Channel 10 or not, okay? Channel 10. There's a program called Dirty Dining where they go to the different restaurant and they check their certificate, health certificate. Okay. I remember one episode where this is a Chinese restaurant somewhere in Miami where he was cited for being rodents and having rodents in his restaurant, and he was supposed to clean them up. I mean, how much effort does it take to pick up the dead rat or a mouse and throw it away? He took the white paint and just painted over the mouse, and you could see the teeth sticking out. And they showed it. I said, why not just pick it up and cook it? Yeah. Just kidding. Just kidding. I think part of that was decomposed, but the teeth was sticking out. He just picked the darn thing up and throw it away. Okay. Nah, let me just paint over it. No one will notice. Okay. How lazy can you be? All right. All right. Third shape, spirochetes. All right. These are the bacteria that live in our mouth, normal flora of our mouth, spirochetes. Not all spirochetes, remember that. Okay, this is one. Okay, this is the shape that lives in our mouth. Couple of things that you need to remember that makes them different from spirula. Spirulas are rigid. These are flexible. Flexible. Spirula, they are motile by polar flagella. Okay, right? These are motile by axial filament. What is it? Okay, let me show you. Which is also called endoflagellum. Endoflagellum. Okay, I think I have it in the. Do I have a picture? Okay, let's first look at these two examples. Borrelia burgdorferi. I know. Yeah. Borrelia burgdorferi. Anyone knows what that causes? Yes, you do. It has something to do with the deer and the tick. There you go. Very good. This is the bacterium that lives in the mouth of the deer tick that causes Lyme disease. Deer tick. Borrelia burgdorferi. And this is the one we already know. Treponema pallidum. Syphilis. Syphilis. Okay. So both of them, they are motile. Okay. They are flexible. But they have this type of flagella. Make a note. Endoflagella. Their flagellum is not free. Do I have a picture here or should I draw? Okay, let's see. Okay. That's spirochete, but don't have a picture of. Let me just save time and show you this instead of drawing. Okay. I think mine is better. If I draw, it's going to be easier to understand, but I'll show you. It's on page 83 in your textbook. Page 83 in your text. Axial filament, endoflagellum. Here we go. This is the real bacterium right here. Okay. Spirochete. And you see this yellow color right here? It's like a rope-like structure that actually wraps around the bacterium. Okay? But it's not free. It is enclosed in a membrane or a sac. Okay? That's why it's called endoflagellum or axial filament. I'll draw my own simple diagram so you'll understand it better. But this is what they're showing right here. You see there's a little brown layer outside. That is called outer sheath. And this filament rope kind of wraps around it. And then you have the bacterium inside. Let me draw my simplified version. That's the bacterium. Now, if this is spirillum, let's do the spirillum first. Spirillum. Where's the flagellum? Here. This end or that end? And it's going to be free, right? This is exoflagellum. Exoflagellum, which is also terminal. Terminal or polar. Terminal or polar. Because it's one end. But if you look at spirochetes, in spirochetes, their flagellum is attached at one end, but it's not free. It kind of wraps around the bacterium like this. And then there's a membrane that encloses it. So this is endoflagellum. Endoflagellum or axial filament or and here's the membrane that surrounds it. And this is called outer sheath. Outer sheath. Outer sheath. Okay? So that's your endoflagellum. By the way, bacteria are motile by these two methods only. Either by exoflagella or there's something called flagella or axial filament. If they don't have either one of them, they are non-motile. They have no other method of locomotion. Remember that for the test. Oh. Size of the bacterium. Oh, before we go to size, okay, that's fine. Let's look at the size. Okay, I'm going to skip the size for a second and then come back to it. There are two two other shapes that are less common. Three shapes are common: cocci, bacilli, and spiral shape. These two shapes, star shape, okay, and square shape bacteria, we don't encounter them every single day. Why? Because these are halophilic bacteria, salt-loving bacteria. You will find them in salty environment. They were actually discovered in, I believe, in Salt Lake, Utah. Okay, star shape and square shape. Halo means salt, philic, loving, salt loving. If you love sugar, what would you call that bacteria? Some bacteria loves sugar. Sacrophilic, sacro from sugar, sacrophilic. Okay, bacteria that love heat, they are thermophilic, thermophilic. Okay. Okay. Can these bacteria, halophilic bacteria, cause human disease? Okay. Okay. This is kind of, you don't have the information that you need to answer this question. So, but the answer is halophilic bacteria, yes. Halophilic bacteria do cause human disease, but not the star shape. Yeah. Not the star shape or the square shape. There are other halophile bacteria. The bacteria that live on your skin, Staphylococcus epidermidis, is a halophile. Is a halophile. What is a halophile? Salt-loving, right? These are true halophile. If you don't have enough salt, at least 10 to 15% they will not grow. But our bacteria that grows in our skin, Staphylococcus epidermidis, it can tolerate up to 7.5. It's not a true halophile. But these are true halophile, or these are called obligate halophile. Obligate means must have. Must have. So these are obligate. So if the question is, can obligate halophile cause human disease? The answer is no. Can halophile cause human disease? Yes. Yeah. Because they don't demand, they can tolerate. What is the normal salt concentration of human body? 0.9, 0.85 to 0.9%. So they can go from 0.9 to 7.5%. That's how much they can tolerate. Okay? That's why if you don't save your potato salads, okay, at the proper temperature, staph will still grow. If you don't refrigerate it, high salt concentration, staph will grow and make the toxin, and it will put you in ER within a few minutes. 15, 20 minutes, you will be in ER throwing up. Okay? Yeah.
Obligate,
Must have.
so they will not cause disease, or
They will not. Yeah. Oh, only in pickled humans, mummies. Yeah. Okay. Just kidding. Okay. If you anybody who has 25% salt, okay. Yeah. All right. Okay. Let's go back. Actually, come on. Come on. Okay.
So where do they cause disease then?
Where do they cause disease?
They can't in humans.
Indirectly. If you have salt, if you have fish that is stored with high salt concentration, they will grow and they will make you sick indirectly.
Is
Okay.
that what causes the sickness in the fish other than mercury? It's another area that...
That's different thing. Completely different. Yeah. Size of bacteria. Okay. The three common bacteria that we encounter, their size. Look at the diameter. All three, cocci, bacilli, and vibrio, same diameter, 0.2 to 2 micrometer. 0.2 to 2 micrometer in diameter. Length, if you remember Anton von Leeuwenhoek, remember Anton von Leeuwenhoek, who fell off from his chair when he saw something moving? Okay. This is the one that he saw moving in his mouth. Spirochete. Look at the length of the spirochete. Wow. No wonder he jumped. And this thing moving in his mouth, look at the size, up to 100 micrometer long, from 0.5 to 100. Okay. Bacilli can up to 8. Of course, cocci, length is not applicable. Okay. arrangement of bacteria. These two terminologies I think I mentioned last time when we were talking about acid-fast stain. Remember? Acid-fast stain? Okay. Most bacteria, they are monomorphic. All cells, all microorganisms in a pure culture, they have same single shape. And most bacteria are like that. But pleomorphism, okay. I gave you two names last time, right? Acid-fast bacteria, mycobacteria and nocardia. Here are some other bacteria, okay, other than the first one. Only the first one is acid-fast. Number 2, 3, and 4, they are non-acid-fast bacteria, but they are pleomorphic. Okay, how many of you see floaters in your eyes? Floaters, lots of us we see. Do they always have the same shape? Never, never. Why? Because this is the bacterium, Corynebacteria, that lives in our eye. Okay, it is pleomorphic. This is the bacteria, okay, that lives in our eye. That's pleomorphic right there. Okay, this is non-pathogenic. Okay, this is called commensal bacterium. Commensal, not communist, but commensal. Okay, commensalism, a relationship in which one organism benefits, but it doesn't harm the other. Commensalism. One is mutualism, where both organisms benefit from each other. One is parasitism. That is all the diseases when one organism benefits and the other one is harmed. Okay, but this one is commensalism. It benefits from us, but we don't get any benefit from it. Okay, but it doesn't harm us. Okay. So these are the bacteria that are pleomorphic. Okay. I sent you a chart last night. I hope you got it. Okay. All these bacteria are on this chart. So memorize those names, please. Okay. Rhizobium, what does this do? One of the most important functions on the planet. Other than decomposition. I know, it doesn't do decomposition. Okay. But next to decomposition, nitrogen fixation. Nitrogen fixation. What is that? Okay. Now, just very quickly, not going to ask you this on the test, just for FYI. Okay. Nitrogen is the most essential part of all proteins, amino acids. Without nitrogen, you cannot make protein, right? No proteins, no life. But most of the nitrogen is found in what form? Gas, nitrogen gas. We humans inhale out the gas, nitrogen gas, and get our nitrogen. It must be converted into organic, right there, organic form, in the form of proteins, so we can consume it. So who does that for us? Bacteria. If you grow your own leguminous plants, like bean plants at home or other legumes at home, pull the bean from the ground, you'll see nodules in the roots. Crush it, put a drop of iodine on it. Observe it under the microscope, and you will see this bacteria. That's called nitrogen fixation. What is it? Conversion of nitrogen gas into nitrate and nitrite. If you buy any preserved food, they will have nitrate and nitrite in it as preservative. In large quantities, it can kill you. But plants, they love nitrate and nitrite. You go to Home Depot and buy nitrate and nitrite as a fertilizer. So what happens? It lives in the roots of the plants. It converts gas to inorganic nitrate and nitrite. So plants eat nitrate and nitrite, and who eats plants? Animals. We eat animals. So that's the cycle called nitrogen cycle. Okay. Now I'm going to ask you. It's called nitrogen fixation. Okay. Yep.
I have a question. Are they preservative, nitrate, but are they cancerous also?
Cancerous? Absolutely. Yeah. Stay away from preserved food if you can. Most definitely. If you consume preserved food, canned food in large quantities, try to avoid them. Yeah. Just try not to live nowadays, okay? Yeah. I mean, everything that you do is cancer causing, really. The water that you drink, it has trihalomethane because our water is cleaned with chlorine. And the chlorine breaks down, you get a chemical in the water called trihalomethane. In lab animals, it causes cancer. Do you have a choice? Those of you who think you are drinking bottled water Okay, is it cleaner than the tap water? Not really. So it's a different debate. I'll come back later. Okay. So everything we do nowadays leads to something. All right. Arrangement of cocci. Coccus singular, cocci plural. Okay. So let me go to the diagram. You have these names. I'll just go to the diagram, read you these names. So come on. Bacilli. All right. Here we go. Pardon me? All right. Bigger? Full screen? Yeah, I can do that. Sure. Mm-hmm. Better? All right. Okay. All right. All right. So, arrangement of cocci. Some cocci, they always grow in pairs. Memorize the names, please, the one that you have in your notes. There are no cocci that grow singly. You may see them on your slide when you do gram stain singly, because when you are making smear, you pull them apart. But there are no cocci that grow singly. Here is a diplococci. There's a bacterium that causes gonorrhea, Neisseria gonorrhoeae, that grows in pairs always. You go to the doctor and the doctor tells you, you have strep throat. This is what the doctor is telling you, strep. Strep means chain. Cocci. Cocci means round. Round bacteria growing in chain. Streptococcus pyogenes. Example. Many of you saw your plates that you streaked last time. Yellow growth, right? Okay. Tetrad. This is the bacterium that grows in tetrad. Always groups of 4. Micrococcus luteus. Beautiful yellow colonies. Micrococcus luteus. And then Sarcina subflava. Okay. Always groups of 8. What type of microscopy is best to observe all 8 cells of Sarcina subflava? What type of microscopy would be ideal to see all 8 cells of Sarcina subflava? Three-dimensional. There's only one that allows you three-dimensional. Phase contrast. Phase contrast is the only one that gives you three-dimensional picture. Yeah. Phase contrast or confocal or interference. Same thing. Yep. Okay. After 8, it is all the same. Staph, S-T-A-P-H, staph. Staph means bunch. When at your office they say, let's have a staff meeting. That literally means let's bunch up. That's what it means. Staph means bunch. Cocci, round. Round bacteria in a bunch. Staphylococci. Okay. Bacilli. Some bacteria, like Pseudomonas aeruginosa, the one that we saw in lab, okay, motility, they always grow in single. Okay, lonely bacteria, okay. Single. Some they grow in pairs. Okay, believe it or not, E. coli grows like this. Diplobacilli. This is the true arrangement of E. coli. And you thought E. coli is cocci. It is so teeny tiny that under the microscope, the magnification that we have, okay, it looks like a cocci. But it's actually a rod, not only single rod, but it grows in pairs. Diplobacilli. This was no problem. We have seen it more than once. Strep means chain here. Again, chain. But this is bacilli. So Bacillus cereus, Bacillus anthracis, Bacillus subtilis, and so on. Identity crisis here. Who am I? Okay, right here. Yeah. Cocobacillus, in between cocci. Okay. Cocobacillus. Not true cocci, not true bacilli. This is the bacterium that grows in our large intestine called Alcaligenes faecalis. Name is in your handout. Alcaligenes faecalis. Like E. coli, it is non-pathogenic, but it could be potential pathogen if it ends up in the wrong part of the body. It can cause disease. Okay? Sure. Pardon me? Alkaligenes faecalis.
And that's in the intestine.
Large intestine. It is alkalophile, alkaline-loving bacteria. Most bacteria are neutrophile, but this bacteria, Alkaligenes faecalis, is alkalophile, prefers to grow at alkaline pH. Yeah. Okay. Memorization, done. Understanding now. All right. Now you should be able to answer a whole lot of your exam. Now we are going to look at a typical bacteria, the different parts, chemical composition, and their shape. Okay. Now, what you're looking at right now, right here is, do all bacteria look like this? No. What you see here is superimposition. Okay. Not all bacteria are rod shaped. Not all bacteria have flagella. Not all bacteria have all these structures. I think I mentioned last time, all living cells, they have three things in common, right? Those are, number one, plasma membrane, DNA or nuclear material, and the cytoplasm. Right there. Okay? Why do we have so many? This is for your next questions for the heck of it. Okay? Why do cells have these many ribosomes? See? Billions maybe. Okay. The granular appearance of the cytoplasm is not because of minerals and all that. It's because of the presence of literally billions of ribosomes. Why so many? Why not 10, 50, 100,000? Why so many? Why so many ribosomes? Partial credit for that, sure. Yeah. That's okay. Okay, fine. Just we'll talk about that next time. All right. All right. So what you see right here is a superimposition, all the possible parts of bacteria. Not all bacteria have all these parts, but we are going to talk about all these parts, their chemical composition and function. First, we are going to talk about extracellular parts, starting with the appendages of bacteria. The first appendage, number one, flagella. Second appendage, fimbri. And third, right here, pilus. So there are three possible appendages that bacteria can have. Flagella, fimbri, and pilus. Then we are going to move on to the layers, these layers that you have. Three possible layers. Plasma membrane, cell membrane, and capsule. Together they are called the envelope. And then we are going to go inside the cell. So let's start with the appendages of the bacteria. Come on. Appendages. Now, three appendages, but look at this one. Appendages for motility. Only one type, right? Flagella. Okay. Exoflagellum. Remember the function of the exoflagellum is locomotion, allows the bacteria to move. Remember, need to memorize this word right here. It is made up of a protein called flagellin, which allows the bacteria to move. Remember the diameter and the length of this. 20 nanometer in diameter, nano. Make sure you underline the... You don't have it?
We don't have this.
Yes.
No, it's just broken all over now.
Yeah, bacteria platform is approximately. I
Page
have about
5?
6. Yeah.
Does it?
Write it down, no problem. Those of you who don't have it or having problem locating it, write it down. Take 30 seconds, write it down. All right.
Are you going to be able to furnish us with the PowerPoint notes, like you said, something that...
Yeah, one of these days before the end of the semester, or you can stop by at my office anytime with a thumb drive and I can give it to you. Yeah, for $50. No, just bring me your thumb drive. Yeah, okay, sure. All right. I know what you're thinking. Why didn't you tell me that this morning? Yeah. Okay. All right. Come on. Go. Okay, fine. Parts. 3 major parts of a flagellum. Everybody found it? Yeah. Everybody has it? Thank you. Okay. Here are the 3 major parts of a flagellum. Okay. Filament, hook, and basal body. Okay. I'm going to show it to you. There are no diagrams on the first exam, so don't worry about the diagram. Do I have? I should have it. There we go. All right. This is on page number 81 in your textbook. Page 81. Okay. Don't draw it. Okay. Draw it. There's no way you can draw that. Okay. Okay. All right. Okay. Just want to label 3 parts. Okay. Here's the filament. Here's the whip-like structure that is outside the cell, whip-like filament. Okay. Hook is the connector, you can call it, that connects the filament to the basal body. Here's the basal body right here, this whole thing. What is basal body? You can call it the foundation. Foundation that holds the structure that holds the flagellum inside the cell. Basal body is the foundation that holds the flagellum inside the bacterial cell. Okay, so those are your 3 parts. Now the movement of flagellum is not like the movement of the sperm. Wiggle, wiggle, wiggle. Okay. Eukaryotic flagella, that's how they move. Wiggle. Okay. But bacteria, okay, they actually have motors. Just like the motor that you have in your car, that actually propels. These rings that you see right here, these are actually motors that actually rotate, and they actually propel the flagella. You see the round motion right there? Okay. It actually, these rings, okay, they turn and it propels the flagellum. So it's not a whip-like motion at all. All right? Now, I'm not going to go into the detail of how many rings, because we have not talked about the cell structure of the cell wall. You see number of rings right here, 4 here and 2 here, right? Why? Because this is gram-negative bacteria, this is gram-positive bacteria. Remember we talked about last time, gram-positive bacteria, they have more what? Peptidoglycan. This peptidoglycan right here, okay? And gram-negative bacteria, they have more what? Lipid. So this is all lipid right there, and they have, you see, when people, non-scientific people, they draw a diagram, they mess up. Okay, look at the amount of peptidoglycan here, same amount here and here. Bad diagram. I will make it much thinner, maybe just a thin layer right here, but that's very bad diagram. Anyways, all right. Just remember 3 parts: filament, hook. All right. Arrangement of flagella. Come on. Numbers and arrangement, it varies. Different bacteria have different flagella. Some bacteria, I think you have something. These are pictures first. Okay, fine. I'll go to the notes later. Let's look at this. Some bacteria, they have only single flagellum at one end right here, the one that I have drawn right here. You see this one right here? That is polar or terminal. This is also called mono. Mono means single. Trichus, I just forgot. What the heck, trichus? Trichus? Hair-like structure. Trichus, yeah, trichus. Monotrichus, one hair-like structure at one end, and polar, monotrichus. Single flagellum at one end. It could be on this end or that end. Right there, monotrichus, single flagellum at one end. Amphitrichus, okay. Now this diagram shows only one flagellum at each end, but you could have more than one flagellum. One or more than one flagellum at each end of the bacterium. That is called amphitrichus. Amphitrichus. Lophotrichus. Lopho means bunch or a tuft. T-U-F-T, tuft, bunch, cluster. Cluster of flagella at only one end. One end. That's called Lophotrichus flagellum. Lophotrichus arrangement. So these three, B, C, and D, you can call them what? Their flagella is attached at end, one end or both end. These are called terminal flagella. Terminal. Terminal, terminal, terminal. Right? Terminal. One end or both end, but they are still terminal. Right? So terminal can be categorized into monotrichus, amphitrichus, and lophotrichus. And then some bacteria, they have their flagella all over. Just all over. This is called peri. Peri means around. Around. Peritrichus flagella. Okay. Another method of locomotion, second type of appendage, okay, axial filament or endoflagellum. Remember this, please. Underline it, highlight. You have these notes. Only found on spirochetes. And remember the examples of spirochetes that I gave you. Two examples, right? And I already explained what axial filament is, called endoflagellum, right? It's like rope or twine that is just kind of wraps around the bacteria. Okay. Texas. What is Texas? T-A-X-I-S. Movement. Yeah, movement. Movement towards a stimuli. True or false? Okay. 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 Movement towards a stimulus is called Texas. True or false? False.
Towards or away.
Okay, yeah.
Either way.
Yeah. Yeah. Could be towards or away. So false. It could be towards or away. If I don't take shower for 3 days, what kind of Texas you will have?
Away.
Away. Okay. so Texas means okay, movement either towards or away. Bugs, you have seen, they are attracted to street lights in summer. Phototexis, right? Phototexis. Another Texas, some people, cruel people, they have these lights in their backyard that zaps the bugs. Bzz, bzz. Okay. Okay. That is also attracted towards. And when you go to the woods or for picnic outside, you put in repellent. What kind of Texas is that? Away. Away. Yeah. Right. So Texas could be movement either towards or away. Directional, either towards or away. Movement of a cell towards or away from a Texas. Okay. That's done. We are done with flagella. Second appendage, Pili or pilus. And the function is conjugation. What is that? What is conjugation? Pardon me? Together. Sticking together. Okay. And the purpose of that sticking together is reproduction. Okay. Is she right? So when two cells stick together, they reproduce. True or false? No, that's not sex. That's not sex pili. Transfer of something. The function of pilus, okay, is to transfer DNA from one cell to another. Okay, transfer of DNA. Yeah. Remember, there is no sex in bacteria. We told that. We talked about that last time. No gametes involved.
So why it's called Flagellar?
Because the function is transfer. Right? Yeah. What is the function of the penis? No, I'm just asking you to transfer the sperms, right? Right? It's just transfer something.
Helix is to transfer DNA from
one cell to another. Or to be more specific, okay, from male to female actually. Now you must be wondering, I just told you there is no sex, so what the heck? Where is this male and female bacteria? So let's talk about that. Okay, actually that is true. There are male and female bacteria. How much time? 10 minutes, so I have to move it a little fast. Okay. All right. Let me just first let you read this. Okay. Long tubular. Okay, the first structure that we looked at, the first appendage that we looked at, flagella, non-tubular. It's not a hose, because this is going to transfer something. So it is hose-like, tubular structure. Okay. It is made up of what? I should have made this red too. Made up of a protein called pilin. Underline it, highlight it, please. Flagella is made up of flagellin, pilin, pilus, pilin. Only found on the surface of gram-negative bacteria. Okay. Now let's see. You have gram-negative bacteria. One is going to be male. Let's call this one male. Male. Okay. No. Male, he has its chromosome. All bacteria have their chromosome. So here's the male chromosome, double-stranded, single chromosome. So bacteria needs this. And here's the female. Let's keep them separate for now. Okay.
This is only bacteria we're referring to, right?
Believe it or not, yes.
The eyelashes, that's the bacterial way.
All right. So female has her own chromosome. Okay. So they both have their own chromosome. So this is female. Why are we calling this male? Because of this little piece of DNA that the female does not have. This little piece of DNA right here, this red piece, which is called plasmid. And what is it? Plasmid. To be more specific, I'm going to call it F plasmid. F. F stands for fertility. F hyphen plasmid. Fertility plasmid. Okay. Plasmid is defined as extra chromosomal DNA. Extra chromosomal DNA. If I take this piece of plasmid from this bacteria, what's going to happen? Bacteria is going to lose its penis. Literally. That's it. So the information for this pilus is located where? Right here. That's why this male is male. Because of that pilus, it can make this pilus right here. That's why it's called fertility plasmid. So when the conjugate, okay, this pilus attaches to the female, and what do you think happens? What is transferred? Copy of the plasmid. Male makes a copy of the plasmid and very happily gives it to the female. And what do you think now female turns to? Male. Sex change operation. Yep. Now, this is male. You can also call it F positive because of the plasmid. F positive. They have F positive, F plasmid. And this is called F negative. No plasmid. F negative, female. But after the conjugation, F negative becomes what? F positive. Yes?
Okay, so there's still really a male and female bacteria. It's just one's F positive and one's F negative.
Very good. Excellent. Yeah, there are no female. Male. They don't make gametes. Just to differentiate between the two, we call them male and female. Because male always give. Poor male. But male always give. Always. But remember, it's not. Sometimes students say conjugation is exchange. It's not exchange. One-way transfer. Transfer of DNA from male to female. Female always receives. One-way transfer. Female benefits. Okay.
Okay, but the female now becomes a guy.
Of course.
Okay, so I guess, like, do we, just eventually run out?
No, listen what happened. Okay, what is the practical use for this, really? Here. Other than F plasmid, here is another plasmid called R plasmid. R plasmid. R stands for resistance. Resistance. Antibiotic resistance. Okay? So let's say male is resistant to penicillin. Penicillin resistant. And female is penicillin sensitive. They conjugate. Male gives a copy to the female. What happens to the female? Penicillin resistant. This is one way how bacteria acquire resistance from other bacteria. And one plasmid can have resistance for hundreds of genes, hundreds of antibiotics. Okay? One more last thing, then I'll stop. Another type of plasmid is called M plasmid. M. That stands for metabolic plasmid. M. M. Plasmid. Metabolic plasmid. Let's say male can go to the cheesecake factory every single day because he can tolerate lactose. He's lactose tolerant. He can digest lactose. Lac positive. Lactose positive. Female is lactose intolerant. She cannot go. Say, I'm sorry, you go by yourself. Okay. So I said, no problemo. Let me give you this gene. It's okay. So he gives the gene to the female. Now she becomes what? Lactose positive. So this is just one example. It could be any physiological reaction. That's why it's called metabolic plasmid. Okay? So conjugation is a process in which DNA is transferred from one gram-negative bacterium to another through pilus. Good day. Okay, sure.